Hello viewers, I am Minhaj Sirajuddin. I am a faculty at Institut, Bangalore. My research interests are cytoskeleton and contractile machinery across scale dimensions with a particular focus on heart diseases and neurological diseases. Our project, together with Professor Carsten Janke from Curie Institute Orsay, the project is called Control of Microtubule Dynamics by Tubulin Code. Now this is a mouthful. I will break it down into simple parts. So we wanted to understand how an important element inside our cells are being regulated. That is the crux of the project title. And if you imagine cell, which is the smallest unit of our human body, there are proteins which actually forms like a network or roads, railway roads, and you can imagine as a city as a cell, and there are railways and roadways, and there where you have trucks and cars and buses and motor vehicles moving in roads, and trains are moving in one direction, and that is what we call as cytoskeleton filaments. It provides a framework for the cell. At the same time, it also provides a framework for transport of molecules across small parts of the cells. I mean, you might think that, like, okay, cell is very small, but the cargo is even smaller, at least 1,000 times smaller, so it needs to go from A to B. So these are the filaments which we call it as cytoskeleton, and microtubule is one of them, which is an important one. Microtubule is involved in, for example, cell division, transport of many of the molecules inside the cell, and many other important functions they perform. And microtubules are also very important target for cancer therapy. There are many drugs which target microtubule cytoskeleton. For example, colchicine, Taxol, and vincristine are some of the famous drugs. Now, if you imagine in humans there are so many different types of cells, and each cell performs a different task, and this cytoskeleton also performs a different task in each different cell. So we have to understand how these are performing in different tasks. So for that, like, we generated a marker, and that was one of the highlight of our work. And this was possible only by collaborating with our French scientists, who are actually a world leader in understanding microtubule cytoskeleton polymer. And we teamed up with them because we had complementary skill sets. Our lab was more interested in biochemistry, structure, and other in vitro related work, and they are more expertise in cell biology, imaging, and also mouse models to study these molecules. So we teamed up, and then we had a lot of exchange. For example, the CEFIPRA funding actually helped us to collaborate, not just collaborate, but also have exchange of people from France to India, and at the same time materials were exchanged between our labs. We were able to send trainees across different labs so that, like, they can train and learn from each other labs and perform experiments. For example, in one case, we sent a lab member to their lab to perform a neuronal experiment, which was not possible for us here. At the same time, they were able to send a trainee, a PhD student, to our lab to perform a structural biology cryo-electron microscopy. In our institution, we had the cryo-electron microscopy facility, so we were able to get the high resolution structures, which was also published. So altogether, it was a mutual, wonderful collaboration. The CEFIPRA funded project resulted in several publications which are high impact, at the same time garnered much visibility. More importantly, we were able to identify a marker which is very well recognized across the world now, and it is available as a resource for other researchers to take up and study in their lab. And we have also patented this molecule for commercial interest. And in future, our collaborations have already set up pathways for exchange in other means. So thank you, CEFIPRA, for funding this project and making a fruitful research collaboration. Okay, so now we have determined that like the binders are binding to the microtubule, right? Now we want to establish what is the specificity of that. So for that we will perform an ELISA experiment, and we will do an ELISA in such a way like how we have been doing the turf microscopy also. So first you layer your BSA biotin, and then you you wash. Then you also add streptavidin, exactly same way as you do the turf microscopy, and then you do another wash. Then you start adding your peptide, which has a biotin, and it will bind to the streptavidin. Then you wash several times. Then you have a plate where you have in each row you will have one type of peptide, and then you take your binders, different binders, and then you incubate them. The final thing would be that like after few minutes or so you do washes.
This, we need to block this.
Yes, exactly. So the blocking is very critical here. So what you can do is you can use the same blocking buffers which you've been using in your turf microscope.
This is Rosetta bacterial competence cell. So we take DNA, like plasmid, whatever we want to express, and like 100 nanogram of DNA is sufficient. And then add to these competence cells. After that, you incubate this on ice for 30 minutes, and then do a heat shock for 45 seconds. And after that you keep it back on ice for like 5 to 10 minutes, and then add LB or SOC media and incubate it at 37 for 1 hour, and then plate it whatever on the plates on LB agar, in which you will have a antibiotic that is like that antibiotic which these bacteria or these cells will have a plasmid which will have that antibiotic resistance gene against it. So then next day you will get the plate with the colony, and then you inoculate those colonies in the LD with the same antibiotics, and then you do a larger culture and do purify your protein from that. So that is the one.
line.
So this is 35 meter. This is why there is no binding.
Oh yeah, I see. There is absolutely no binding. It looks good. Yeah, that's great.
Then there is
like shown one binding, and so you have at least like 58% eliminated from this process, right?
yeah. So this is something.
So this is somewhere intermediate.
They have something which is like
Oh wow.
yeah. This is meter to
61. Great. So now you should just do the specificity experiments with ELISA and also do unmodified microtubule then.
yes.
Yeah, great result.
So I tried to do that HEK experiment.
Mm-hmm.
But the protein had disappeared. That was the lysate.
No, but you need to do it with purified protein. The HEK. Sorry, the HEK microtubule.
Yes. So we are doing this transformation Rosetta today, and maybe tomorrow we'll, 96 will be. Day after tomorrow we'll do the sonation, and maybe if we get the time, we can do that.
Great. Yeah, that would be a good result to have.
Yeah.
Long live India and France friendship.